Hello, and welcome to Teach Me in 10. In today's episode, we're discussing an increasingly important topic in food safety, sample preparation for PFAS analysis. We'll take you through the essentials of sample prep, exploring techniques like catchers and micro-SPE and how they help to ensure accuracy. Joining us is Dr. Hagen M. Gegner, Scientific Communication and Project Specialist at CTC Analytics AG. Dr. Gegner will show us how automation is transforming these workflows, boosting data quality, speeding up processes, and helping labs deliver safer, more authentic food products.
Hi everyone, and thanks for the kind introduction again. Today we're diving into the world of PFAS analysis in food. We'll explore how we tackle these persistent compounds, talk about sample prep, and how automation can help us. So, what exactly is the challenge with PFAS? Well, imagine a chemical bond, the carbon-fluorine bond, that's incredibly strong. The stability makes PFAS fantastic for things like non-stick pans and fighting fires, but it also means they just don't leave. They've earned the nickname forever chemicals for a reason. Consequently, industrial activities, consumer product use, and historical pollution have led to them being pretty much everywhere in the soil, water, in the air. If we go through them, the primary sources of soil contamination includes the application of biosolids, compost, and wastewater irrigation, as well as the use of PFAS-containing pesticides. For water, aqueous film-forming foams, AFFF, used for firefighting are a major source of PFAS contamination in water bodies. And in the air, well, that's a little bit understudied. PFAS can also be transported through the atmosphere, contributing to their global distribution. But again, more research is needed here. Naturally, if they're everywhere in the environment, they're going to find their way onto our plates. In agriculture, this is a growing concern. They can be absorbed by crops from contaminated soil or water, or accumulate in livestock. Think dairy, meat, eggs. Here, the highest concentrations are found in animal products. In aquatic ecosystems, you would find PFAS concentrating up the food chain, especially in fish and other seafood. Here, the highest concentrations are found in top predators due to their bioaccumulating characteristics. And let's not forget direct contact, so leaching from those nice grease-resistant wrappers. Processing equipment can also add PFAS directly to food. So summarizing, they're quite adept to infiltrating our food supply. How are we handling this growing problem in terms of regulations? Well, if we look at the EU, they are taking a notably broad approach. They are setting binding limits for PFAS sums in drinking water, already have limits for 4 key PFAS in certain foods, and are rolling out a sweeping ban on PFAS in all food packaging that will start in late 2026. Meanwhile, the US federally focuses its binding limits on 6 PFAS in tap water via the EPA with compliance by 2029. But for food and food packaging, well, the FDA doesn't set federal limits. It relies more on monitoring voluntary industry phase-outs and leaving many states to create their own packaging bans. It's a bit over a regulatory patchwork there. However, they do provide methods with minimum detection limits and limits of quantification that we will include in our analysis later on. This divergence means that analytical labs need methods that are not just sensitive, but also very adaptable, and not just in their sample prep, but also to their sample types. Luckily, we have a nice toolbox for this. You will recognize many of these workhorses in our PAL system universe, but in sample prep in general. Catchers, various flavors of solid phase extractions, and so on. Picking the best method or tool isn't straightforward, though. It's a balancing act on the food matrix complexity, which PFAS we are after, how low we need to go, and finally, how many samples we need to go through, as well as is it acceptable with the regulations you may or may not follow. Let's look a little bit deeper at those sample prep methods. You have liquid or solid liquid extractions, very classical one that you can also miniaturize. Here I linked a dispersive liquid micro extraction that is also automated. You have SPE, a very common and powerful cleanup and or concentration method. This can be done manually offline or fully automated online with columns and valve switching or using tiny micro SPE cartridges. Then you have catchers borrowed from our friends from the pesticide residue analysis. It's a clever combination of solvent extraction and salting out phase separation, followed by a quick and dirty cleanup. In the past, it was usually dispersive SPE, but now recently, this is also more pushed into the direction of micro SPE with these cartridges. You also have SPME. This you would usually find more in the GCMS realm, but it can also be used for LCMS analysis. Based on a recent review, it's interesting to see that most of the labs use a combination of offline SPE with an LCMS platform. Picking up on that, we now would like to go a little bit into more detail and look at a real-world example combining the very popular catchers with micro SPE on an LCMS platform with our PAL system. Here we tackle 73 PFAS in shrimp. This isn't just automating one step, it's the whole process. So spiking standards, the catchers workflow, mixing, centrifugation, the part of the micro SPE cleanup, as well as the online injection. The nice thing is here that while you are measuring one sample with your LCMS system, the PAL system already preps the next one, meaning you're really having some kind of walk away automation setup here, including the analysis part. Automation is great and all, but does it actually deliver? On the right side, you see in a nutshell a few numbers that I pulled from the app node. You can also see on the graph, the red line here is the FDA's analytical limits from their published method for the determination of 30 PFAS in food and feed using LCMS. You can see we're continuously below that line for our method detection limits as well as LOQs, and that is great. But it's not just about sensitivity, the accuracy and reproducibility as well as repeatability was good too, with recoveries typically between 65 to 135% for most of the 73 PFAS across the QC levels, and even tighter ones, 80 to 120 for the regulated PFAS. Repeatability was also very good, below 19%, and often much better, below 12% for the key regulated PFAS. So you can see this is a ready to be implemented workflow, fully automated. So if we now zoom out a bit, I think it becomes clear that accurate PFAS analysis in food is vital, but clearly complex. Currently, most of the studies focus on established offline SPE LCMS methods, although this can be automated and is already automated and arguably would make it more robust and more routine, it's still underutilized. I showed you guys a catchers combined with micro SPE example that provides consistent and nicely reliable data needed for the PFAS we know about and regulate today. However, the PFAS story isn't finished. We need non-targeted methods to find unexpected PFAS, and perhaps the nightmare of any analyst, we need constant vigilance against background contamination from our own lab environment. There, it's important to recognize that you can upgrade your systems, you can take care of these tubings with peak tubings, or maybe switch your valves and, vials with PFAS-free consumables, for example, to counter this background contamination a little bit more. That brings me to the end of this whirlwind tour of automation and sample prep and PFAS analysis in food. Thanks for sticking with me. Feel free to reach out, and thank you for your attention. Ciao, ciao.
Today's episode is brought to you by PAL System, the modular automation platform for LCMS, GCMS, and beyond. Our system helps labs run smarter, not harder, helping to enhance efficiency, reproducibility, and data quality. With automated solutions, scientists are better equipped to tackle the complex food safety and authenticity challenges, delivering the reliable results that protect public health and consumer trust. Thank you for joining us for another Teach Me in 10. Be sure to subscribe so you never miss out on more bite-sized science insights.